Welcome to the Deep Dive. This is where we take those really dense research papers, the ones that usually need a PhD and maybe a lot of coffee, and we pull out the core ideas just for you.
Making the complex accessible?
Exactly.
Hmm.
So today, we're diving right into the real cutting edge, where engineering meets biology. We're talking neuromorphic electronics, bioelectronic interfaces.
Deep tech territory.
It really is.
Yeah.
Our source is this, well, pretty groundbreaking paper from Nature Communications. And our mission today is, I think, quite specific. We want to unpack how scientists finally built an artificial neuron that doesn't just simulate a biological one, but actually mirrors its fundamental operating physics. We're talking energy efficiency here.
Right. That's the kicker.
Yeah, because for decades, people have been trying to copy the brain, right? But hitting this wall when it comes to matching the actual electricity, the signals, the power used.
Mismatched parameters, basically. Totally. So the breakthrough we're looking at today, it seems they've crossed that final hurdle. They achieved parameter matching, amplitude, energy, timing. And this apparently allows for seamless real-time connection with living cells.
And that seamless part is so critical.
Yeah.
Because, biological neurons, they're whispering. They use these spiking action potentials with incredibly low amplitude and energy.
How low are we talking?
We're in the picojoule range per spike. It's just astonishingly efficient. If our electronics are shouting, electrically speaking, you just can't have that real conversation with the body. You overwhelm it.
Okay, shouting versus whispering. I like that. Let's get into it. All right, let's unpack this gap then. Before this paper, we knew low power was the goal. But just how bad was the mismatch? What was the big functional bottleneck stopping older artificial neurons?
Well, it was fundamentally about power and voltage compatibility. Think about it. A real neuron fires with signals maybe 70 to 130 millivolts, right? MV.
Yeah, millivolts. Really subtle. But the artificial devices we were building, usually with silicon or, other standard materials, they needed 0.5 volts, sometimes way more, just to operate.
Okay, so 0.5 volts versus, say, 0.1 volts.
Yeah.
That's 5 times, maybe 10 times higher.
Exactly, an order of magnitude, easily. You're essentially trying to interface delicate biological systems with something that's, electronically speaking, much louder, much more powerful. It limits density, safety, everything for implants.
So it wasn't just the circuit design tweak needed. It sounds like a fundamental material problem.
That's right.
So what was the material innovation here? What let them finally bridge that gap?
It came from, well, a pretty unexpected place, memristors, but built using protein nanowires.
Protein nanowires.
Yeah, and not synthetic ones initially anyway. They're harvested from a microbe called Geobacter sulfureducens.
Hold on, a microbe. Like bacteria are making parts for advanced electronics.
Yeah.
That sounds a bit sci-fi.
It does, doesn't it? But it's quite elegant, actually. See, this microbe naturally produces these protein wires on its outside. It uses them to basically exchange electrical charge with its environment, like little biological wires.
Okay.
And what makes them perfect is their size. They are incredibly thin, just 2 to 3 nanometers in diameter.
Oh, tiny.
Really tiny. And that nanoscale structure allows you to build stable electronic devices that just inherently operate at these very low voltages, the ones that match biology.
Okay, I get the principle, but I have to push back a bit on practicality.
Yeah.
Harvesting proteins from one specific microbe. How scalable is that if you're thinking future medical devices? Is this just a lab trick?
That's a totally fair question.
Yeah.
And yes, this specific paper used harvested stuff for the proof of concept, but the underlying material, it's a protein. Proteins can potentially be made synthetically or, produced in bulk using biofermentation, like how we make insulin.
Oh, okay.
The real takeaway isn't necessarily how they got it this time, but the physical principle that a nanoscale protein device can switch reliably at bio-friendly voltages.
Right, so what were the numbers? Did they actually close that voltage gap?
They did. The first big win was consistent switching. Their protein nanowire memristor, it switched on and off around 60 millivolts.
Okay, 60 milliV. That's right in the biological range you mentioned.
Exactly. And the current was tiny too, about 1.7 nanoamps, both right where they need to be. And crucially, they measured the resistance when the device was off.
And that matters because?
Because it was about 200 megaohms. A megaohm is a million ohms, so very high resistance.
Okay, 200 million ohms. What's the significance of that number?
It's significant because it's really close to the electrical resistance of an actual cell membrane, which is typically in the 50 to 300 megaohm range.
Oh, I see.
So they didn't just match the switching voltage, they also matched the sort of passive electrical property of the cell boundary itself. They'd laid the groundwork.
Okay, this is where it gets really interesting for me. Matching static properties like resistance, that's one step. But a real neuron is dynamic, right? It adds up inputs, fires, and then critically it has to reset itself. That refractory period.
Yes, the reset is key.
And previous designs often struggled there, needing external circuits just to force that reset. So how did this device go from just switching once to firing continuously, like a real neuron, with that all-important self-reset?
They managed it with a surprisingly, almost deceptively simple setup. They integrated their protein memristor with a basic RC circuit.
Okay, RC circuit resistor, capacitor, we hear that a lot. Let's just quickly define what the R and the C are representing here in this biological mimicry.
Good point. So the capacitor C essentially acts like the cell membrane. It stores the electrical charge.
Okay.
The resistor R is designed to mimic the slower, stabilizing flow of ions, specifically like potassium channels in a biological neuron, which help reset things.
Got it. And the memristor itself.
The memristor is the star. It mimics the fast voltage-gated ion channels, like the sodium channels that cause the spike.
Okay, so walk me through the cycle. An input signal comes in.
Right. First phase, integration. The input causes basically atoms to physically accumulate inside the memristor's switching filament. This is like the neuron gathering charge until it hits its trigger point.
Building up until the threshold.
Exactly. Then, boom, depolarization or firing. Threshold hit, the memristor flips to its on-state low resistance. This lets charge rush onto the capacitor really quickly. That's your spike, your action potential. It mirrors how sodium channels fly open in a real neuron.
Okay, the spike happens. Now, the tricky part, the reset. How does this RC circuit make it turn off by itself? No external timer needed.
And this is really the clever bit, the core breakthrough, phase 3, repolarization and the refractory period. So the capacitor is now fully charged, right? It holds a relatively high voltage from the spike. Well, that high voltage on the capacitor actually imposes a reverse voltage back across the memristor. It pushes back, electronically speaking. And that reverse bias is enough to force the memristor to automatically switch back to its off state, its high resistance state.
So the act of firing itself triggers a shutdown.
Precisely. It's self-limiting. Biologically, it's like how the high membrane potential during a spike causes sodium channels to automatically close to inactivate. This artificial neuron does it passively using the capacitor's charge. Then, the charge on the capacitor slowly leaks away through the resistor, R.
The potassium channel analog.
Exactly, completing the reset, readying it for the next input.
Okay, but wait, if you need to charge that capacitor up significantly to get that reverse bias voltage, aren't you just, like, spending energy charging the capacitor instead of using external reset circuits? Is it really that much more efficient overall?
It's a massive net gain, yes.
Yeah.
Because the RC part is passive. There's energy to charge the capacitor, sure, but it avoids needing complex active control circuits like CMOS logic, which would constantly consume power, probably nanowatts or even microwatts, just sitting there managing the timing.
Right, those control circuits add up.
They do. This passive reset mechanism lets the entire thing operate at incredibly low energy levels.
So what were the final numbers on efficiency? You mentioned picojoules before. Did they hit that target?
They absolutely nailed it. The output spikes reached about 120 millivolts, smack in the middle of the biological range.
Okay.
And the energy, they measured spiking energy as low as 0.2 picojoules per spike.
0.2.
PJ.
Yep. And the biological range is generally considered 0.3 to 100 PJ.
Yeah.
So it fits perfectly. It's truly biorealistic energy consumption.
Let me just pause on that 0.2 picojoules. For you listening, that's like a trillionth of the energy a tiny LED might use in a second. The efficiency difference is just mind-boggling. It really could change what's feasible for devices inside the body.
It absolutely could. And functionally, it wasn't just the energy. The artificial neuron also showed that nonlinear behavior you see in biology, where firing frequency increases with stronger input, but then it kind of saturates, hits a limit around 60 hertz or so in their tests.
So it matched the physics and the functional behavior?
Sure. Okay, so they've built this incredibly efficient, biorealistic, artificial neuron. But, knowledge isn't much use unless you apply it. So beyond just firing like a neuron, what other, maybe more complex biological functions could they start to emulate with this device?
Right. The next logical step was neuromodulation. This is a huge part of how the brain works. It's not just electrical signals. It's the chemical environment around the neurons that tunes their activity. Think hormones, neurotransmitters, the whole chemical soup.
Okay, so how do you get an electronic circuit to respond to chemistry?
Remember the RC circuit. The resistor, R, was the potassium channel analog controlling the reset time.
Yeah.
They engineered it so that the value of that resistance could be changed by external chemical sensors. So change the chemistry, change the resistance, change the reset time.
And therefore change the firing frequency of the artificial neuron.
Clover.
Exactly. It lets the chemical environment directly modulate the neuron's electrical activity.
So what chemicals did they test this with? They started with something fundamental. Sodium ions, Na+, just basic electrolytes. They used a type of chemical sensor called a POT.PSS chimera-resistor.
POT.PSS chimera-resistor. Can you quickly unpack that? What is it?
Sure. It's basically a resistor made from special conductive plastics, polymers, that are known to be pretty biocompatible. And crucially, their electrical resistance changes measurably when the concentration of certain chemicals around them changes.
Got it. So what happened with sodium?
They found that as they increased the sodium concentration within the normal range you'd find in the body, 0 to 150 millimolar, the resistance of this sensor went up.
Okay.
And because this sensor was acting as the R in the RC circuit, that increased resistance made the artificial neuron fire more frequently.
Which matches biology.
Perfectly. Higher sodium concentration generally makes real neurons more excitable, easier to fire. So check one for neuromodulation mimicry.
Okay, that's a nice linear relationship. But I understand the second test was more complex. They used dopamine.
Yes, dopamine, a key neurotransmitter, obviously involved in reward, learning, movement. And dopamine signaling in the brain is famously nuanced. It often shows what's called an ambipolar profile.
Ambipolar, meaning?
Meaning its effect isn't just more dopamine, more activity. It depends on the concentration and which receptors it hits. Generally, low dopamine levels can increase excitability, often via D2 receptors, but high levels can actually suppress excitability, acting through D1 receptors.
So it's like a push-pull effect depending on the dose.
Exactly. It's a critical balancing act in the brain.
How on earth did they replicate that with their artificial neuron?
They swapped out the sensor. This time they used a graphene-based chimera-resistor, known to be sensitive to dopamine. And remarkably, this graphene sensor showed the exact same ambipolar response to dopamine. Its resistance first decreased at low concentrations, then increased again at higher concentrations.
Wow, the sensor itself mirrored the biological effect.
It did. And because that resistance was controlling the artificial neuron's firing, the neuron's firing frequency also showed an ambipolar trend. It fired faster at low dopamine than slower at high dopamine. They replicated that complex modulation, about a 25–34% change in frequency.
That's really powerful. If that ambipolar dopamine response is so vital for, say, cognitive functions, like deciding whether to stick with something known or explore something new.
Explore, exploit trade-off, yeah.
Right. Does replicating it in this simple low-power circuit mean we could actually use this to model, maybe even diagnose issues related to dopamine signaling? What's the near-term relevance here?
I think the relevance is potentially huge. That dopamine balance is fundamental to learning and decision-making. Being able to model it accurately, chemically, and electronically with something this energy efficient. It opens doors for building better diagnostic tools or maybe much more sophisticated drug screening platforms.
Platforms that react to subtle chemical changes, more like a real neuron would.
Exactly. You could potentially screen drugs for neurological conditions much more effectively, or maybe even personalize treatments based on subtle chemical signatures.
Okay, so let's pull back to the big picture again.
Yeah.
They've got this efficient neuron. It can even respond to chemistry. But the ultimate dream of bioelectronics is always about bridging that gap. Right. Blurring the line between electronics and biology.
Just seamless integration.
Yeah. Did they actually test if this artificial neuron could, like, connect to and interpret signals from living cells in real time? A genuine two-way street.
They did. This was the crucial final demonstration. They built a sensing system using a flexible mesh, kind of like a tiny electronic net, and they embedded this mesh directly into living heart tissue grown in the lab. Specifically, cardiomyocytes derived from human embryonic stem cells, or HESC-CMs.
Okay, wait. Heart cells. Why not use actual neurons for the big finale?
It's a good question. Cardiomyocytes, heart muscle cells, are actually excellent models for this kind of test. They generate really strong, regular, easily interpretable electrical signals when they beat.
Makes sense. Easier to measure and verify the connection.
Exactly. It gives you a very clear, reliable biological signal to work with. You know for sure if your sensors are picking it up correctly and if the artificial neuron is responding as expected.
Okay, so they have the heart tissue beating, the mesh sensors picking up the electrical signals in real time. What did they do with those signals?
They fed those live signals directly in real time into their protein nanowire artificial neuron.
Right. And the critical test wasn't just if it could respond, but how it responded.
Precisely. The test was designed to see if the artificial neuron could act as a kind of real-time physiological state detector. Could it tell the difference between the tissue behaving normally and the tissue being in a different state, say, affected by a drug?
Okay, how did that play out?
So in the normal state, the cultured heart tissue was beating relatively slowly, firing electrically at about zero point 0.4 hertz, less than once per second. At this slow rate, the input spikes were too far apart. The memristor in the artificial neuron would start to accumulate a bit of charge from a spike, but then it would leak away before the next spike arrived. Net effect, no accumulation, threshold never reached.
So the artificial neuron stayed quiet. It listened, but didn't fire.
Exactly. It remained silent, effectively recognizing the normal state.
Then they changed the state.
Yes. They introduced norepinephrine. It's a drug sometimes used for heart failure, known to increase heart rate and excitability. This put the cells into a drugged state.
And the tissue started beating faster.
Right. The firing frequency picked up to about 0.6 hertz, still not super fast, but critically faster than before. And now, with spikes coming closer together...
The memristor didn't have time to leak.
Exactly. The dynamic shifted. Now there was net accumulation of charge in the memristor filament with each spike. It reached the threshold, and the artificial neuron started to fire.
Wow. So it successfully distinguished between the normal and the drug state based purely on that subtle frequency shift from the live cells.
In real time. It wasn't just processing recorded data, it was interpreting a live physiological change.
That feels like a really significant step.
Yeah.
But playing devil's advocate again, sticking electronics, even a flexible mesh, into living tissue. What about the long term? Biocompatibility, immune response, potential damage even with low power? Isn't that still a huge hurdle for any implanted device?
Oh, absolutely. That's the perennial challenge, no doubt. And the researchers acknowledge it. Any foreign object carries risks. However, several things make this approach promising.
Okay.
First, the materials themselves, the protein nanowires, the graphene, are chosen partly for their potential biocompatibility. Second, using a flexible mesh geometry is much less mechanically stressful on soft tissue than rigid implant. And third, the ultra-low power operation is key. Less power means less heat, less electrical stress on the surrounding cells.
Reducing potential damage vectors.
Exactly. It's not a solved problem, but this work represents a really big step towards creating devices that the body might actually tolerate long-term, moving away from those bulky, power-hungry electronics of the past. Hashtag, outro.
So wrapping up, I think there are really two headline achievements here. First, they genuinely seem to have closed that decades-old gap, the parameter mismatch in voltage, energy, amplitude, using these clever bio-derived protein nanowires.
Matching the whisper, not shouting.
Right. And second, they showed it wasn't just about matching static numbers. They demonstrated complex dynamic functions that mimic biology, like that ambipolar chemical neuromodulation, and importantly, interpreting real-time signals from living cells to detect a change in state.
Yeah, that real-time connection feels critical. So this research, it really demonstrates that building these ultra-energy efficient, bio-friendly neuromorphic systems, it's feasible. We can potentially operate electronics right down at the physical level of biology.
Seems so. And it leaves you thinking, if we can now build artificial neurons that perfectly mirror the dynamics, the chemistry response, and crucially, that picojoule energy use of real neurons, what fundamental questions about our own brains, about how we compute so efficiently, might this technology finally help us unlock?
It opens up entirely new ways to probe those questions.
It really makes you wonder, doesn't it? Maybe the path to truly powerful next-generation computing isn't just bigger chips or faster clocks. Maybe it's about figuring out how nature runs these incredibly complex systems on the electrical equivalent of almost nothing. Could the blueprint for the most powerful computers actually come from? Microwave's whisper.